Hello everyone, I'm Mojtaba and welcome to another episode of A Sip of Chemistry. In this journey of meeting amazing people who have shaped the field of glass science and magnetic resonance, both of which are my interests, I had the opportunity to speak with Peter Hore from Oxford University. He started his career with studying magnetic resonance and the effects of electrons and nuclear spins on chemical reactivity, which is known as spin chemistry. However, in the past few years, more specifically since 2005, exploring bird migration through magnetoreception concept has been his main focus. He was elected as a Fellow of the Royal Society and has received numerous awards for his valuable contributions to science, including the publication of hundreds of articles and several books. Please stay tuned and watch my interview with Professor Peter Hore. Thank you so much, Professor. Thank you for agreeing doing this interview. I'm so excited about it. So at the beginning, as usual, I would love to ask you about your education, your academic background. When and where did you get your degrees?
Okay, well, I grew up in Cornwall in the southwest of England. My final 2 years at high school, I studied mathematics, physics, and chemistry. In 1973, I went to Oxford University to study chemistry. That was a 4-year course, and the final year was entirely research. I did a research project on time-resolved EPR, supervised by Keith McLaughlin. At the end of that year, I then started my PhD, also in Keith's group, and that was more EPR of short-lived free radicals. In 1980, I postdoc in the Netherlands at the University of Groningen in the group of Robert Kaptein, and that was my first exposure to NMR. After that, I came back to Oxford for a 3-year junior research fellowship, and that was in association with Ray Freeman. So more NMR, but after a year, I got the position that I currently have in Oxford, and I've been here ever since.
Why did you choose chemistry as your major when you wanted to go to a university?
I think what got me hooked on chemistry at the beginning was the periodic table. I mean, it's an amazing construct, organizing the properties, the electronic structures of what seemed to me at the time as a young teenager, completely mystifying array of elements. And the fact that you could systematize their properties and the periodicity, the relations of one element to another, I found that fascinating. As a young scientist, I suppose I already knew I was going to be a scientist, even at the age of 11 or 12. I also had very good teachers at school who inspired me. And although I did consider other degrees, in the end, I plumped for chemistry, and I certainly haven't regretted that.
Okay, amazing. So then you did your undergrad study. What motivated you to do EPR, to join your supervisor's research lab?
So Keith had given us lectures during the first 3 years, many other Oxford professors, and I enjoyed those very much. Some of those were on magnetic resonance. I guess I liked quantum mechanics at that time, and so this was a way of combining quantum mechanics with spectroscopy. And also Keith, I had come across in person while trying to do an EPR experiment in the undergraduate teaching labs in Oxford. And this was a machine that contained thermodynamic valves. It was before printed circuit boards, and my partner and I were struggling to get this machine to work, and Keith came and showed us how, and then told us about the exciting things that were happening in his lab using EPR. And so, yeah, it was an obvious choice when I had to find a supervisor for my final year research project.
Okay, yeah, that was my next question. How did you approach your supervisor? So it was a research course, you did something about it, and actually you made your decision. Up until 2005, I guess, you mainly focused on NMR spectroscopy and EPR. Uh, so at that time, what was your main concern? I mean, which questions you were trying to answer using NMR spectroscopy?
There were a number of things. I mean, it wasn't entirely NMR. So I got hooked on spin through the EPR, electron spins, nuclear spins, and that's been really the theme of my whole research career. So up to about 2005, we did NMR of proteins, protein folding. We were trying to develop new methods for studying protein folding in real time, using spin chemical principles. I was also doing spin dynamic simulations in relation to photosynthetic energy transfer. Radical pairs are formed photochemically in photosynthetic reaction centers, and I was interested in simulating EPR spectra in order to extract more information on the initial electron transfer steps involved in harnessing solar energy. We were also interested at that time in effects of weak magnetic fields on chemical reactions. One of the contexts for that is the possibility that man-made electromagnetic fields might be harmful to human health. And so we were trying to learn more about how weak magnetic fields might interact with chemical reactions.
So would you tell us, is it harmful or not? To which degree?
Well, we don't know. I think it's unlikely. I mean, the kind of magnetic fields we were interested in at the time were mains frequencies, so 50, 60 hertz. So this is clearly not ionizing radiation. There are mechanisms by which, in principle, such low frequency electromagnetic fields could affect chemistry, but the fields themselves, emitted from electrical equipment, for example, are really extremely weak. And I think it would be remarkable if anyone could show convincingly that there really is an effect on biology, which, after all, contains so many mechanisms to protect against free radical damage.
Yeah. So you mainly did, in the case of NMR again, you did solution NMR, right? Have you done any solid state NMR?
No solid state at all, I'm afraid. Nothing on glasses either, which I know is your research interest.
Nuclear magnetic resonance, that's the title of one of your books. So at that time, you wrote two books, I guess, on on resonance spectroscopy, magnetic resonance spectroscopy. One of them, nuclear magnetic resonance, which I like it. It's actually easy to follow, understandable, and here in our university, in Department of Chemistry, it's used as a reference, a textbook for undergrad students. So what motivated you to write this book?
Okay, I'm glad you like the book and that your department is using it. What motivated me? Well, my colleague, Richard Compton, in physical chemistry in Oxford, was one of the three editors of the Oxford Chemistry series, Oxford Chemistry Primers. So these are published by Oxford University Press, and Richard and two others had been recruited as editors of the series. And so they committed to finding 100 people to write primers. And Richard asked me to do the NMR one. And prior to that, I hadn't considered writing a textbook, but it seemed like a good idea. I'd already given a lecture course to Oxford undergraduates on NMR. And so I thought it wouldn't be too much effort to produce a textbook. I think I was wrong in that. It took a lot of work, but I quite enjoyed doing that. And yeah, that's how the book came about.
Again, I would like to ask about NMR spectroscopy, then we move to your recent research interest. What do you see the most important characteristic of NMR spectroscopy? I mean, what makes it a powerful tool for studying materials?
Well, I mean, there are all sorts of things. There are so many different nuclei one can study. You get information on structure, molecular dynamics, on chemical reactivity. I mean, it's extraordinarily versatile and powerful spectroscopic technique. And NMR spectroscopists have proved to be incredibly inventive in thinking of new experiments, new applications, new ways of using magnetic resonance, NMR, EPR, and related methods to learn about molecules, their chemistry, physics, biology. I mean, what's not to like about it, really? Difficult to pick out anything in particular.
I know that a Nobel Prize was awarded to Professor Robert Richard Ernest. I guess he worked on the resolution of 2D NMR, increasing the resolution of 2D NMR. So, beside that work, what do you think that has revolutionized the NMR spectroscopy?
Yes, well, I mean, one of the earlier discoveries or inventions of Richard Ernst, which really revolutionized NMR, was the idea of using pulsed radio frequency fields and Fourier transform methods to obtain the spectrum. I mean, that revolutionized really the practice of NMR. The improvements of sensitivity were so enormous, of doing pulsed excitation, excite and detect all the spins in the sample simultaneously, rather than sweeping the field slowly through resonance and detecting each peak one at a time. And then that opened up all the possible NMR pulse sequences that are used today, including the two-dimensional and higher dimensional methods you've just mentioned. That they clearly had an enormous impact on the way that NMR is done in all sorts of areas. I think one of the most exciting areas I see at the moment, and I'm a bit out of touch because I haven't been to NMR conferences for 10 or 15 years now. My research has taken a different direction, but clearly one of the most exciting areas at the moment is dynamic nuclear polarization. The ability to transfer magnetization from electron spins to nuclear spins and enhance detection sensitivity in that way. There seems to be an enormous range of different ways of doing that and a large number of different applications. And that seems to be something that huge numbers of people are working on now. And I think that looks to an outsider like me really very exciting now.
Yeah, I think that's the future of NMR spectroscopy. DNP could be one of the way that could help answering some of questions that were impossible maybe a decade, 2 decades ago. But eventually you stopped running NMR. Was it boring?
No, it wasn't boring. I just got interested in other things. It must have been in the early part of the century that we started to work on the animal navigation. So I had worked on spin chemistry, radical pairs, effects of magnetic fields and magnetic interactions on chemical reactions, as I mentioned earlier, for most of my career. And so I had been aware of a paper published in 1978 by Klaus Schulten suggesting that the mechanism by which birds detect the Earth's magnetic field might rely on spin chemistry, that is, reactions with radical pairs as transient intermediates. And we, like most other people, didn't take a great deal of notice of that paper because it was quite abstract. I think it's a brilliant idea, very far-sighted. But at the time, Schulten wasn't able to suggest a molecule in which all this magnetically sensitive chemistry could take place inside a bird's body. And also, the paper was quite technical, quite mathematical, and that, I suspect, put off the biologists who might otherwise have found it an intriguing idea. And then all that changed in 2000 when Schulten suggested that cryptochrome, a protein that had only been discovered in the early 1990s in plants and had subsequently been found in many other organisms, including vertebrates, birds, humans even. By the end of that decade, it was clear that cryptochrome had at least some of the properties that might be suitable as a magnetoreceptor. And so Schulten published that in 2000, that suggestion. And it was that got us and others interested in this possibility. And so gradually we started to work on that, initially by doing some simulations, spin dynamic simulations of radical pairs in cryptochromes, and then started some experiments. And of course, that wasn't NMR because we were interested in weak magnetic fields. The Earth's magnetic field is 50 microtesla. That's quite a bit smaller than most of the magnetic fields that one would use for NMR or EPR. And so that research into the mechanism of the magnetic compass in birds grew and took up more and more of our time. It turned out to be a fascinating topic. And so all the other stuff kind of got pushed to one side. And so that included the NMR and some of the other things we were doing at the time. And then I guess by the end of the 2000s, 2010 or so, it was the major research topic that we were working on. And so for sure, we didn't get bored or fed up with NMR. It just kind of got pushed out of the way by other interests.
Yeah. So magnetoreception, would you describe it briefly? What does it mean and how you measure it? I mean, you said you cannot use EPR or NMR. How you measure the properties that you are interested in?
Yeah, so, well, first of all, how do we know that birds have a magnetic compass? Well, that's something that had been suspected of homing pigeons, certainly in the 19th century. But it was really demonstrated convincingly only for the first time in the 1960s by Wolfgang Wilczko in Germany. And he and his supervisor put European robins, so small migratory songbirds, into magnetic fields. And they showed that if you did these behavioral experiments during the migratory season in the spring or the autumn, then the birds would, if you confined them to orientation cages, under conditions where the only directional information they had came from the magnetic field that you provided them with, then the birds, and this had been well known, the birds during the migratory seasons, towards the end of the day at dusk, when the sun was setting, this is when the birds would normally set off to migrate. They fly at night, these small songbirds, and they exhibited a behavior known as migratory restlessness. So they became agitated during the migratory seasons, preparatory to setting off to fly during the night. And if you do this behavioral test in orientation cages, then the direction in which they jump in their attempt to escape from these cages is the direction in which they would fly if you released them. So all you need to do to show that they have a magnetic compass is to test them in a magnetic field that has been rotated, say by 120 degrees. And if they then jump in the appropriate direction, shifted by 180 degrees, 120 degrees, then you know that they've got a magnetic compass, that they use that compass to orient themselves. So those were the original experiments, and they're still done that way to this day. So that's the way that we can test whether the birds are using a magnetic compass. And we know now that many migratory songbirds do use a magnetic compass to navigate. And you can see why that might be useful. Some of them fly thousands of kilometers and need to know which direction to fly, and also need to know where they are during those journeys. So the experiments we do, so I'm a physical chemist, I don't do these behavioral experiments. I have collaborators who are biologists who are extremely skilled in doing that kind of thing. The experiments we do are of various sorts. One type is on the proteins. I mentioned cryptochromes, on the proteins that we think are the magnetic sensors. So Shulton, as I said, suggested cryptochrome as a possibility 24 years ago, and it's still really the only candidate we have. It's the only known photoreceptor that forms radical pairs when it absorbs blue light. And we know blue light is involved because the birds can only use their compass if they are either subjected to blue light or they have a blue light component in the white light under which you test them. And so the cryptochromes are the only proteins that form radical pairs when they absorb the kind of wavelengths of visible light that the birds need for their compass to work. So we study cryptochromes in the lab, purified cryptochromes, and then use various methods of optical spectroscopy, both absorption and fluorescence, to monitor the photochemical reactions of these proteins in magnetic fields. And in that way, we've been able to show that these proteins have at least some of the properties that you would need for a magnetoreceptor protein. And we know that the cryptochromes are located in the right place in the bird's bodies, that is, in the retinas, the layers of photoreceptor cells at the back of the eye, which are involved in vision. We also do experiments, my collaborators and I, with model compounds which bear very little relation to the structure of the proteins, but also form radical pairs when they absorb light. And some of these model compounds are easier to handle than the proteins, and so allow us to learn more about the mechanism by which radical pair reactions could be sensitive to very weak magnetic fields.
These questions comes from my limited knowledge on proteins. Do you develop or do you make these proteins in the lab, or you take it from the bird?
So all the proteins come from the lab of my collaborator in Oldenburg in Germany, Henrik Mauritzen. So he's a biologist. He has a large biochemistry lab, which really is a protein factory. He set up a very efficient and productive factory for producing all kinds of different cryptochromes. And it really is a wonderful resource. We email his people and say, we would like this mutant of this cryptochrome, and almost always they're able to produce it for us within a month. So we get large quantities of highly pure proteins, both wild type and mutant, bird proteins mostly, but also some from other organisms like plants. And these are produced in bacterial cell culture. So we take the genome of the birds, some of which migrate, some don't. We like to compare migratory and non-migratory species. We take the genetic information from the birds and then get E. coli to overexpress these proteins, which can then be separated and purified.
Okay, amazing. And also, you have worked on Drosophila, if I'm right about it. You recently published an article and you challenged the results of another research group, and I think not the original researcher. Another research group, they replied and they commented and they defended the original work, and you responded. Would you tell us more about it?
Yes, so we were interested in whether Drosophila would show magnetic field effects that might also be cryptochrome mediated. So fruit flies, Drosophila, have just one cryptochrome. They're simpler organisms than birds. And that cryptochrome has a great deal of similarity to the bird cryptochromes, or at least the one of the 6 known bird cryptochromes that we think is most likely to be a magnetic sensor. So, and also the literature has quite a number of papers stretching back over 10 or 20 years from a variety of research groups who have done behavioral studies on fruit flies and other insects and have reported magnetic field effects on their behavior. So the idea, and this is a collaboration with, again, Henrik in Oldenburg, the idea was that we would first of all repeat some of these measurements just to verify that we could observe magnetically sensitive behaviors in fruit flies. And then we could use the wonderful genetics available for fruit flies to make all kinds of mutant flies, mutated at different positions in their cryptochromes, so that we could learn more about magnetic sensing mechanisms. And we were always aware, even at the beginning, that of course flies are not birds. And so anything we learned about the flies would not necessarily be transferable to the birds, but flies are easier to study than birds. We wouldn't be restricted to the migratory seasons. The assay I described to you earlier only works for a couple of months in the spring and again in the autumn. Flies, you don't have the ethical considerations that you have for studying wild birds. And so we thought, and also being smaller, we wouldn't need 2-meter magnetic field coils. We could use perhaps centimeter coils, which would be much easier. So there were very good reasons why we wanted to see these magnetic field effects and expected to see them in fruit flies. And so we tried to replicate studies of research groups that have been published in nature group journals, so in high profile, high impact journals. One was a tea maze, so you give the flies a choice. They can either choose one arm of a maze that is exposed to a magnetic field, or they can walk in the opposite direction where there is no magnetic field. And the original assay tested either naive flies or flies that had previously been conditioned to associate the magnetic field with a reward. And they had reported statistically very significant effects of the flies being attracted or repelled by the magnetic field. So that was one of the assays. And another one used a natural behavior of the flies called negative geotaxis. So this is an escape behavior. Flies naturally tend to climb vertical surfaces. So I'm sure we've all seen flies on our kitchen windows, and typically they climb upwards. And this is an escape behavior. They go towards the light, hopefully away from predators. And so there had been a group in Leicester who had used this assay, flies climbing inside transparent tubes. And they had found that the speed at which they climb could be influenced by magnetic fields that were only 10 times stronger than that of the Earth. So those were basically the two assays that we tried to replicate. And we tried to do this incredibly carefully. And we also tried to do it as closely as possible using the conditions of the original studies. And so the authors of those original studies were very helpful. They either provided the blueprints for the construction of the tea maze, or they allowed us to borrow the equipment they had used for the negative geotaxis. And we also developed our own negative geotaxis assay that we thought might be a bit more sensitive. We were testing flies climbing individually rather than groups of flies. So we had 3 separate assays, and we tested a number of different conditions, 3 or 4 different magnetic field strengths. We tested different wavelengths of light, some of which we expected to activate cryptochrome, some longer wavelengths were not expected to affect cryptochrome. We studied 5 or 6 different strains of Drosophila, some of them supplied by the authors of the original studies. And in total, we studied more than 100,000 fruit flies. And unfortunately, disappointingly, we weren't able to find any statistically significant magnetic field effects. And this really was a great disappointment, because it then made no sense to do any of the experiments we had planned using genetically modified flies. And we were lucky to be able to publish those negative replication studies in a good journal a couple of years ago. And then that prompted, not surprisingly, the authors of the studies we had tried to replicate to write comments, which were also published in the journal, and we responded to those comments. And the conclusion we came to was that the original studies, in our view, were false positives, that inappropriate statistical methods had been used to analyze the data. And also, we felt that our experiments had been done a lot more carefully than the original studies. Now, The original authors disputed that. And so, well, we have the comments and our response now published in the same journal as our negative replication study. And so it's all out there now for people to form their own view about all of this. So we've never claimed that fruit flies have no magnetic behaviors. All we say that is we were unable, after great effort, to find magnetically sensitive behaviors using the TMAs and the geotaxis assays. And that makes us doubt whether the flies have magnetic responses. But we're happy to be proved wrong if other people can show that there was something wrong with our experiments, or that with a different assay, different groups can independently replicate the same experiments. So that's our position with the flies at the moment.
Thank you. Thank you for your explanation. I've not seen, at least I have not seen, such discussions between researchers in the field that I'm actively involved in. I mean, glass science, NMR, and I think it's a good way to improve it. There are some works in the literature that they might be misleading, if researchers do not pay attention.
You're right, it doesn't happen very often. I think it should happen more frequently. It's been, unfortunately, an aspect of this field of magnetoreception that some of the reports of magnetic field effects on animal behavior haven't been able to be replicated independently in different labs. And I think this is because animal behavior is difficult to do carefully, reproducibly, but it is crucially important for the field that we can say with confidence what is reliable, what is not. And so I think it's very important that the journals that publish the original studies also publish negative replication results in cases where there have been careful negative replication studies. I think it's also important for those same journals to publish these kinds of discussions between authors of studies that don't agree with one another. I think that's the way we make progress in science, and in areas where replication has proved to be difficult. I think that is really quite important.
Yeah, exactly. I agree. Um, on your webpage, I've seen that you have raised several questions that, to a large extent, it shows that what's the trend in magnetoreception research interest. But it would be great if you could actually mention some of the big questions you have in mind and would like to answer them in the future.
Yes. So that there's one thing which I would really love to get to the bottom of. So it turns out, and I think this is really quite remarkable, that the birds can be prevented from using their magnetic compass by radio frequency electromagnetic fields. So when they're tested in the assay that I described to you earlier, in the orientation cages, when they jump in appropriate directions, depending on which magnetic field you apply, those magnetic fields, of course, are static. Like the Earth's magnetic field, they're 50 microtesla in strength, and they don't vary in time. Now, it's clear, and this has been replicated in a number of different research groups now over the last 10 or 20 years, that if the birds are at the same time exposed to weak radio frequency magnetic fields, with frequencies in the range from a few megahertz up to a few hundred megahertz, then they can no longer use their magnetic compass. So instead of jumping in the direction in which they would fly if they were released, depending on the season, spring or autumn, they jump in random directions. So they have no longer any sense of direction from the Earth's magnetic field. And this, even though those radio frequency magnetic fields are 1,000 or more times weaker even than the Earth's magnetic field, which is itself pretty weak. Now, we know from laboratory studies of organic radical pairs that radical pair reactions can be sensitive to radio frequency fields, either on their own or in combination with static fields. And this is a resonant effect. So the radicals, which are the sensors, have internal magnetic interactions between the electron spins and nuclear spins. And these hyperfine interactions, of course, usually give rise to structure in EPR spectra, hyperfine splittings. And the strength of those hyperfine interactions typically in the low megahertz frequency range. And so the way in which radical pairs respond to these very weak magnetic fields is that the radical pairs can exist in two spin states, either singlet or triplet, depending on whether the electron spins, one in each of the radicals, have either anti-parallel spins, that would be a singlet, or parallel electron spins, that would be a triplet. And so when a singlet radical pair, for example, in the cryptochrome, is formed by the absorption of light, followed by electron transfer reactions, you form a radical pair in a singlet state, and then it interconverts coherently with the triplet state. So it's a quantum mechanical superposition state, and it oscillates coherently at megahertz frequencies between singlet and triplet and back again. So if you apply an electromagnetic field at one of those frequencies, so in resonance, then you can do a form of EPR spectroscopy. Okay, you're hitting the frequencies of hyperfine interactions, but then using the bird's behavior to detect resonance instead of the usual electronic methods. Okay, so this you can think of as a form of EPR, but using the bird's behavior as the readout. And so some people like to call this animal detected magnetic resonance. Okay, so you can do that for organic free radical reactions in solution in the lab, where you know what the radicals are, so you know what frequencies ought to be affected. And you can do this kind of spectroscopy, and it works very well. And theoretically, you can understand why it works. But you need those radio frequency fields to be strong enough. And that means typically of the order of 100 microtesla, maybe even 1 millitesla, in order to have this resonant effect. So the big question is, could the same thing be responsible for this disorienting effect on the birds? And at first sight, you would say no, because those radio frequency fields are 100, 1,000, 10,000 times weaker than theoretically you would predict they would need to be to have this resonant effect. Okay, so in principle, there could be a radio frequency field effect that would explain these behavioral observations and consistent with a radical pair mechanism for the magnetic compass. So the way to test this then would be to predict frequencies that should or should not affect the birds, and then test using a behavioral experiment to see whether the birds can still use their compass or not. Okay, so we think that cryptochrome is the right magnetoreceptor, and we think we know what the radicals are inside the protein formed by absorption of blue light. So we know what those radicals are, and so we can measure or calculate all of their hyperfine interactions. So we can then predict the maximum frequency that could be in resonance. And so that will be, so that's a simple quantum chemical calculation. You set up a spin Hamiltonian for this radical pair using the known hyperfine interactions in these two radicals, and then you diagonalize it with a computer to find the eigenvalues, the energy levels of this spin system, including about 20, 30 nuclear spins coupled to one or other of the electron spins. And then you look for the highest and lowest eigenvalue. So the highest energy level and the lowest energy level. And the gap between the two, that will be the highest frequency that could be in resonance with an electromagnetic field. Any higher frequency would be off resonance and should have no effect. Any lower frequency would almost certainly have an effect if the field were strong enough, because if you've got 20 or 30 spins all coupled together, you've got hundreds of thousands of energy levels. So there's almost certainly going to be something in resonance with any lower frequency. Okay, so we can predict what that frequency is for the birds if cryptochrome is their magnetoreceptor. And then Henrik's people in Oldenburg can do behavioral experiments. And so far, those experiments are entirely in agreement with these quantum mechanical predictions. If you use frequencies below the predicted cutoff, then the birds are disoriented, they can't use their compass. If you use a frequency above the cutoff, then there's no effect. They can use their compass perfectly well. So it looks like, even though quantitatively we can't explain this effect because the RF fields really are very weak, qualitatively, it all seems to be consistent. We can do a simple quantum calculation in the computer, and that allows us to predict how the birds behave. And that, I think, is remarkable. And so what I would like to understand is how this really works quantitatively. So is there something in the birds that we really know nothing about, something that makes them much more sensitive to these radio frequency fields than we think they ought to be? So to put it another way, is there something in the theory of the radical pair mechanism, which is now 40, almost 50 years old, something that everyone has missed for all those years, that can make radical pair reactions in a bird's retina much, much more sensitive to time-dependent electromagnetic fields than we think they ought to be? Now, if the answer to that is yes, and we could get to the bottom of that, that would be fascinating from a physics point of view, and it could be hugely important from a biological point of view, because there's no obvious reason why a bird should be abnormally sensitive to very weak radio frequency fields. I mean, the time-dependent magnetic fields do occur naturally, but they contain no useful information about direction. The birds wouldn't benefit at all from evolving sensitivity to radio frequency fields. In any way, the strongest electromagnetic fields at these frequencies that birds are exposed to are man-made. They come from our electronic equipment. And electronic equipment of that sort has only been used for the last 100 years, since we had mains frequency power in our homes and laboratories and industries and so on. And 100 years is too short a time for birds to have evolved a sense sensitivity to those magnetic fields. So if this really is a real effect, and it's a radical pair effect, then it's probably a symptom of extreme sensitivity to the Earth's magnetic field. So this amplification mechanism, if that's what it is, whatever its origin, might be the clue to how this magnetic compass is so incredibly sensitive to magnetic fields of the strength of the Earth.
Yeah, that's amazing. That's a big question, and let's see in the future how your research goes and how you can answer this question. Another question that you raised is it possible to design cheap and versatile organic sensing devices based on the research that you have done, based on your finding? Is it possible, and where is the application if it goes well?
Yeah, I mean, this is not something we work on, but it is something, of course, we've thought about and speculated on. So there are organic spintronic devices that do show magnetic field effects, and the physics of at least some of these are essentially identical to the physics of the radical pairs in cryptochromes. So, for example, in some organic light-emitting diodes, not so far removed from the kind of OLEDs in our mobile phone displays, electron-hole pairs can show effects of weak magnetic fields that have the same origin as the radical pairs in the cryptochromes I've been talking about. So the physics is basically the same. And so a couple of research groups have published papers on these organic semiconductors subject to quite weak static and in some cases radio frequency magnetic fields that show the same kind of effects. So I think this has potential for applications. The problem about studying the proteins is that you can't modify them too much before they stop working. So I said earlier that Henrik's group are extremely proficient in making mutant cryptochromes. That is, they can swap out one of the amino acids that we think is crucial for this magnetic sensing property, and they can put a different one in its place. And almost always that works. But of course, there are only 20 naturally occurring amino acids, and there's a limitation, there's a limit on how many of these substitutions you can make before the bacteria refuse to express the protein, or it proves to be impossible to purify, or the protein won't fold. But of course, organic chemists are hugely inventive about making molecules, and they're not restricted to 20 amino acids or 4 nucleic acids. They can use the whole range of organic chemistry. And it does need to be organic. There are, of course, many, many semiconductors, inorganic materials, but for the most part, as soon as you have heavy metals, then you have strong spin-Orbic coupling, and that means fast spin relaxation, and that very often kills these magnetic field effects from radical pairs. So we usually need organic radicals, so carbon, nitrogen, oxygen, without heavy atoms in order to see these effects. So I think there's enormous scope for making sensors that are inspired by the biology, but which use chemistry, organic chemistry, and also use as a detection, a readout mechanism, either electrical signals, measuring the conductivity, for example, of semiconductor devices, or as in OLEDs, using light output luminescence as a monitor of these magnetic field effects. And there are probably a variety of applications there for measuring the presence, strength, direction of weak magnetic fields. I think once it becomes clear just how versatile and sensitive these devices can be, I think the applications will follow.
Well, let's move to beyond the lab. I would like to ask some other questions related to your personal aspect of your life, if you don't mind. So, what do you do at your spare time?
Well, yeah, I mean, I don't seem to have very much of it at the moment. All the stuff I've been talking about keeps us quite busy, but family, of course, is hugely important. And then I like hiking and cycling. So we've had some wonderful holidays in North America and in Canada in recent years. Hiking around Jasper and Banff, for example, and in national parks in the US. Also hiking and cycling in the UK, not such dramatic landscapes as North America, but still pretty challenging and exciting for walking and cycling.
Yeah, I've been in Banff. Yeah, that's amazing. I love Alberta, Rocky Mountains, Jasper. Yeah. So when you were a kid, what was your dream job?
My dream job when I was a child. Well, I don't know whether it was a dream or not. I mean, I had various ideas at different ages. At one point, I just wanted to be a mathematics teacher in a high school. What else? Oh, yeah, I had an idea about being a forensic pathologist. So I quite liked the idea of being the person who, identified the cause of death. Perhaps in a criminal case, maybe. I suppose one of my hobbies at the moment is crosswords, and maybe that's kind of like a crossword. It's a puzzle, a problem to be solved that could be a mixture of science and intellect. I never got very far with that. I wanted to be a doctor more generally at one point. I'm sure I wanted to cure cancer. Quite a grand ambition, didn't get very far with that, but I'm sure people also had that as an ambition when they were young. And also even when I was applying to universities, I was never quite sure about whether it should be chemistry or biochemistry. In the end, I plumped for the chemistry, which I think was right. But as it turned out, I've done things related to biology, at least the last 20 years or so. So I've kind of come back to that maybe towards the end of my career. So I'm not sure whether that was a dream job, but I mean, those were my early ambitions, I guess.
Yeah, you have followed one of your ambitions. Yeah, you have pursued it. So if now, if let's say there is a parallel world, you can pick a job. Which job now you would like to choose in another world, another life?
In another life, yeah, okay. So this is pure fantasy. I would like to do something artistic. I would like to be a great artist or a musician, a great musician. So, I mean, I have no particular talent in either area, but, you know, I enjoy art, painting, I enjoy listening to music. You know, I mean, one of my favorite painters is Vermeer, the Dutch artist at the end of the 17th century. You know, we only have about 30 paintings of his, and every one of them is a work of genius. You know, if I could be someone whose art was still revered in 300 years' time, that would just be unbelievable. And music, wouldn't it be wonderful if one could be a concert pianist or clarinetist? Or you could write music like Bach, you know, the partitas for solo violin, or that, you know, that wonderful clarinet glissando at the beginning of Rhapsody in Blue. You know, that would be my fantasy. Completely, totally unrealistic. But, you know, if you give me a fantasy world, that's what I would try.
Who knows? Maybe there is another life.
Who knows? You can only dream.
Yeah. Do you have any regret in your life, in your professional?
Regrets? No, not really. I've been incredibly fortunate in all aspects of what I do, both personal life and scientific life. So no regrets. Very happy with where I am.
Great. And the last question, who is your favorite scientist in the history?
Scientist, yeah. I don't know. I think it would have to be someone like Paul Dirac. So, Nobel Prize at the age of 31. You know, the first person to write a PhD thesis on quantum mechanics. You know, a genius theoretical physicist by anyone's standards. You know, even Einstein thought he was wonderful. You know, such insight, such beautiful physics at such a young age, and a very strange man. There's a wonderful biography of Dirac by Graham Farmiloe. It's called The Strangest Man. And he was a very peculiar man. He was introverted. He was very strange in his behavior, a difficult man to engage socially. But my word, was he brilliant. I really recommend this biography. He's a fascinating man. I would have loved to have met him.
Yeah. Well, great. Thank you so much, Professor. Thank you for your time, and it was my pleasure having this great conversation with you. And have a great evening.
Thank you very much. I've really enjoyed it, and have a great day yourself.
Thank you.
Goodbye.
Bye.